Okay, well, let us begin. But before I do that, I am going to pour myself. Yeah, you can mix a little bit of frozen orange juice and add it to your white wine. Regardless, I am going to continue my rabbit hole of this GPLD protein. So here I showed this article the other day where they talked about how upregulating GPLD1 in mice had shown the effects of, how do I phrase that, improving the neurons of older mice. And the way that it worked was older mice, um, if they were sedentary, basically they start getting neurodegenerative diseases as they get older, as you'd expect. However, if they work out, then you end up preventing that from happening. And one of the proteins that is upregulated due to working out is this GPLD1 protein. So GPLD1. So it increases in the blood of mice following exercise. And it is also correlated with the mice's improved cognitive function. Now, correlation doesn't mean causation, right? But what they specifically did here was upregulate this GPLD1 in mice with or without exercise and see if there was an effect on the neurons. And in fact, there was. There was a noticeable significant change in the cognitive function of these older mice. So this would be one of the mechanisms by which something could be synthesized in one organ, this protein for instance, and then it ends up benefiting the cognitive function in a completely different organ, right, the brain. They argued that this GPLD1 is a liver enzyme and it is upregulated in the liver and it's secreted by the liver. However, it appears as though the liver is not the only place where you can get this GPLD1 synthesized. It could also, I've been reading a little bit, it could be synthesized in the islets, or the what's it called? The pancreatic beta cells, or the islets of Langerhans, I think is the pronunciation. Regardless, those are the insulin-producing cells. So what this group was arguing was that it is synthesized by the liver and then it gets out into the rest of the system, and this is how you see some of these beneficial effects. However, I will argue that it might not necessarily be all coming from the liver. It could be synthesized in some other cells as well. So let us begin with my tangent, where I went over this article called The Responsive, or I read over this article, or I should say I read over the abstract of this article. I don't have access to it, and I want to, so I probably will find a way to look at it later. But it's the response of serum glycogen-4 levels and its potential regulatory mechanism to endurance training in chamomile flowers hydroethanolic extract in streptozotocin nicotinamide-induced diabetic rats. So a lot of words there, but I'll come back to this. So this is one paper, and then here we have a different paper called The Endurance Exercise Training Restores the Diabetes-Induced Alterations in Circulating Glycosylphosphatidyl Inositol Specific Phospholipase D level. So this is the GPLD1 protein, right? So this is the same thing. And then I also went over this article called Glucose and Insulin Regulate Glycosylphosphatidyl Inositol Specific Phospholipase D. So let's drink. What is this saying to me? Well, anyways, I'll go over this one as well. Just so you know, there are 3 papers that are going over this. Let me collect my thoughts. So I will go through this paper first, or this abstract first, and then give you my thoughts, and then we will progress to the 2 other papers. Okay, so Glypican-4 is a novel adipomyokine that enhances insulin signaling. So what is glypican-4? So this is a chemical that, or sorry, it is a protein, sorry, that is encoded by the GPC4 gene. It's a cell surface heparan sulfate proteoglycan. Just note, important thing is that it's on the surface of the cells, and it's composed of a membrane-associated protein core substituted with a variable number of heparan sulfate chains. So these heparan sulfate, these are glycosylations that can take place on the surface of these cells. So don't worry too much about that. So the members of the glypican-related integral membrane proteoglycan family, also known as GRIPs because we love our acronyms, these contain a core protein anchored to the cytoplasmic membrane through a glycosyl phosphatidyl inositol linkage. Okay, so this is similar. This is a protein that is anchored to the cellular membrane, right? And these proteins may play a role in the control of cell division, growth regulation. So the GPC4 gene is adjacent to the 3' end of the GPC3 and may also play a role in this particular syndrome. Okay, so don't worry too much about all that. Just know that this is glypican-4, and I should also mention that this is found on the X chromosome. So theoretically, if there was deletions or if there were mutations in this gene, they could be inherited, or I should say it might have gender-based pathologies. Regardless, let's keep on going. So glypican-4 is a novel adipomyokine. So an adipo means fat tissue, like adipose tissue, and myo means muscle. So this is an adipomyokine, which are cell signals that are secreted by the muscles, if I'm remembering correctly. So let's look up. So yeah, a myokine is one of the several hundred cytokines or other small proteins and proteoglycan peptides that are produced and released by muscle cells, or the myocytes. So yeah, these are adipomyokines are, and I believe adipokine, and adipokines or adipocytokines, these are secreted by adipose tissue. So this is a novel adipomyokine, so something that is secreted by at least one of the adipose cells or the muscle cells, and these enhance insulin signaling. So if you have decent amounts of glypican-4 in your system, this is playing a role in the proper insulin signaling. So glycosyl phosphatidyl inositol specific phospholipase D, also known as GPLD1, which is the protein that I was talking about earlier, which when upregulated is associated with better cognitive function. This protein is thought to release GPC4. So remember, GPC4 has this, it is anchored using glycosyl phosphatidyl inositol. So let me get up a photo of, okay, well, it does not have a photo here, but I will go to phosphatidyl inositol and show you. So here we have a phosphatidyl inositol. So this is an inositol group right here, which looks a little bit like a sugar, but it's not exactly quite. Here's the phosphate group, and here is the acyl groups. And so you have two acyl groups, which is a fatty portion of it, and this anchors to the cellular membrane because the cellular membrane has a fatty inside. So this will stick into the cell membrane and anchor whatever's covalently attached to it to the cell membrane. Okay, so frequently you will have glycation that takes place at this inositol group, which will link it to a protein in some way. So remember that this glypican-4 has that anchor on it, and so this GPLD1 will is a phospholipase. It will break up that anchor so that it releases this GPC4. anyways, GPLD1 is in of itself an insulin regulating enzyme. So the beneficial effects of exercise training in chamomile flower extract, also known as CFE, are shown through activation of the Ppar gamma. So which is a promising drug target in diabetes and associated with GPC4 synthesis. So what is Ppar gamma? This is a receptor. So this is called the peroxisome proliferator activated receptor, and this is how they work in general. So let's say you have a diet in adipose tissue, or sorry, you have a diet that has some sort of fatty compound, or this could also come from the adipose tissue, which is your fat tissue, and you could get these fatty acids or fatty components coming in, right? And so this fatty component will interact with one of the Ppar proteins. So here this is Ppar alpha, but here you can see Ppar gamma. So what happens is these compounds will, these fatty acids will come in and they'll interact with the Ppar alpha, and then this Ppar alpha will interact with this RXR, which is the retinoic, I forget the exact name of it, the retinoid X receptor. So don't worry too much about this. This is just another protein that is needed for the for these PPARs to interact with the DNA. So here we have a fatty acid can go and interact with the PPAR alphas. But what also can happen is that a fatty acid could be converted into some form of hormone. And here, this would be an example of the prostaglandins or the leukotrienes, just certain fatty hormones can come in and then interact with the Ppar gamma. And then since this is fatty, it can cross the cell nucleus pretty easily. And so a Ppar gamma will then interact with the RXR, similarly to how the Ppar alphas interact with RXR, and then these will initiate or promote certain gene transcription. So the extract from a chamomile would presumably, well, there are compounds in chamomile and a lot of other, what would the word be? A lot of other plant material, organic compounds that are in these plants will have chemicals in them that can interact with some of the proteins in your body. And so for chamomile extract, it is interacting with this Ppar, or some of the chemicals might be interacting with this Ppar and initiating transcription of particular genes. So let us go to the nomenclature and the tissue distribution. So here we have the alphas, and these are expressed in the liver, kidney, heart, muscle, adipose tissue, and others. We have the betas and the deltas, and they are expressed in many tissues, but specifically the brain, adipose tissue, and the skin. And then we have the gamma, and this is transcribed by the same gene, but this PPAR is through alternative splicing is expressed in 3 forms. So I should say this gamma protein has its own gene, but it can be spliced in different ways depending on what tissue it is in to create 3 completely different proteins with similar functions but come from the same DNA. So this is one of the miraculous things about the human, or I should say DNA, is that the same gene can actually encode for a lot of different proteins based on how the mRNA is spliced differently. So here for gamma 1, this is in all tissues, including the heart, muscle, colon, kidney, pancreas, and the spleen. Gamma 2 is expressed mainly in adipose tissue. So if you say wanted to develop a drug that would target adipose tissue, perhaps you wanted to act upon this gamma 2 receptor, or that would be if you wanted to develop a drug that would attack adipose tissue while leaving other tissues alone, that would be something that you might want to consider. And so here we have gamma 3, which is expressed in the macrophages, large intestine, and the white adipose tissue. So if you wanted to, say, attack white adipose tissue directly, you could use something that would throw a wrench into the gamma 3 version. Regardless, just know that these are These are proteins that will interact, they're transcription factors. They will initiate transcription of the DNA if they get their proper signal. So these chamomile flower extract is thought to interact with the Ppar gammas, and this is a promising drug target in diabetes and associated with GPC4 synthesis. So this Ppar gamma, when it gets activated, it will activate, or I should say, it will cause the transcription of many different proteins in the body, or many different genes will get transcribed, and one of those genes is the GPC4, which will be synthesized as a result of this becoming activated. So this study investigated the effects of 14-week treadmill running and the chamomile extract on the serum GPC4, the GPLD1, and the insulin levels in streptozotocin nicotinamide-induced diabetic rats. So what is streptozotocin? So this is something that looks a little bit like a glucose molecule. So here we have this sugar molecule that looks pretty similar to glucose. However, it's a little bit different in that it has this entire group on it rather than an alcohol group. And it is particularly toxic to the insulin-producing beta cells of the pancreas in mammals. Okay, so this is given to induce diabetes in certain animal models. So, and it is also used to attack cancers that come from the pancreatic islet cells, because since that particular cancer is going to be susceptible to this molecule much more than the normal tissue, that would be a way that you could help kill those cells. Granted, you might induce diabetes as a result of that chemotherapy, but it would be better than dying in many cases. So here is the mechanism. I think that would be important to know. So streptozotocin is a glucosamine nitrosourea compound. So this right here, this is a urea group, and then this right here is a nitroso group. And so this is nitrosourea, and you can see it here where you have your glucosamine, because it's aminated glucosamine, and then this is one side of the urea, and then this is the nitroso group on the other side, and you also have this additional methyl group. Regardless, it's a methylating agent. Or they call it an alkylating agent, which is when you add alkyl groups to certain things in the body. So a methyl group is a type of alkyl group, just keep that in mind. So because of this, it is toxic to cells by causing damage to DNA. So methyl groups have, if you have methylating or alkylating agents, they can be particularly damaging to DNA, because DNA has a lot of groups on it that are very vulnerable to methylation, and once that methylation happens, it can render a lot of that DNA very unable to be transcribed and duplicated correctly. So it needs to be repaired for the DNA to function again. So, though other mechanisms may also contribute, they say. So the DNA damage will induce activation of the PARP. So don't worry too much, but which is likely more important for diabetes induction than the DNA damage itself. So streptozotocin is similar enough to glucose to be transported into the cell by the glucose transfer protein GLUT2. Okay, so glucose doesn't just flow into the cells. Glucose requires transporters to cross the cell membrane, and so you have multiple types of glucose transporters in the body. So glucose 2 is unique in that it is the predominant glucose transporter of the islet cells of the pancreatic beta cells, or the pancreatic what else are they called? Oh, islet cells. Sorry, not beta cells. So it is, since they express so much of this transporter, this molecule will preferentially go into those cells versus the normal other body human cells. I don't want to call them normal, but the different cells in the body that have GLUT1 transporters, for instance, they are going to have more resistance to this molecule. So, so this explains its relative toxicity to beta cells. Oh yeah, it is beta cells, since these cells have relatively high levels of GLUT2. Okay, so what these people are doing is that they will give this streptozotocin nicotinamide, which I don't know if the nicotinamide is doing something else, if it's an adjuvant of some form. Regardless, this is the main toxin, and this is killing, or at least causing, significant disruption in the beta cells or the islet cells. It's creating large disruption in their mechanisms or their cellular signaling, right? It's destroying DNA. It's activating a completely different set of proteins are being transcribed that normally would not have been transcribed due to the protective agents that are being created. So that happens. So which induces diabetes. So methods. So 32 of these streptozotocin nicotinamide-induced diabetic male Y-star rats were randomly assigned into 4 groups: the control group, the training group, the chamomile flower extract treatment, and the training plus the chamomile flower extract treatment. Okay, the training groups were exercised on a treadmill for 5 days per week, and the treating groups were fed with 200 milligrams per kilogram of day of this chamomile flower extract in drinking water for 14 weeks. So finally, the serum GPC4, the GPLD1, and the insulin levels were analyzed through a sandwich ELISA. So, and just remember this GPC4, this is the membrane-anchored protein that gets catalytically released from the cell membrane by GPLD1. So when they're checking it in the serum, it is no longer bound to the cell membrane. It had to have been released in some way, right? So, and then they're also looking for GPLD1 to see if that is increased, and they're looking at insulin levels as well. So here are the results. So compared to the control group, which remember the control group also had, the control group also got poison. They also got the toxin, so they also have diabetes. What would have been nice is if they had another group of rats that did not get the toxin in them, but c'est la vie. So here we have the serum insulin levels were significantly higher in the treated and the training, the chamomile flower extract, and the training plus the chamomile flower extract groups. So this means that insulin there was more of it being released if they were training properly, or if they were taking this chamomile flower extract, or if they were doing both. And as we can see here with the p-values, that the p was less than 0.05 for the training, it was less than 0.05 for the chamomile flower extract, but it was less than 0.01 for when they did both. So this means that there was it was less likely to come out by chance. So while the OGTT, and this is a test, OGTT is a glucose tolerance test, so it's a gold standard for making diagnoses of type 2 diabetes. And so if I don't know all the exact things, but it's basically, I believe, have people fast, give them glucose, and see how their blood glucose changes as a result of that. So the OGTT and the serum GPLD1 levels were significantly lower in the training, the chamomile flower extract, and the training plus chamomile flower extract group. So they ended up having better tolerance, and their GPLD1 levels were a lot lower. So I thought GPLD1 was supposed to be beneficial, so you would want to have more of it. However, it's lower in the training groups, the CFE groups, and the TCFE groups. What the hell is going on here? So, and some of the other papers, I'll go over what I think about that later. So, changes. So basically, these GPLD1 levels are going down when they train or when they take CFE or when they train with CFE. And this is not insignificant. This is P of less than 0.001. So it is very statistically significant that the GPLD1 levels and the glucose sensitivity levels are, or I should say the glucose resilience levels, are going down. And I meant to say glucose sensitivity. Regardless, changes in the serum GPC4 levels were not significant. So there was not a lot of change in the GPC4. So that's interesting. So I thought up here, yeah, so they were looking for GPC4, but apparently there's not a significant difference in the groups. Interesting. So the GPLD1 levels were negatively correlated with insulin levels. And HOMAIS, which this is the homeostatic model assessment for assessing beta cell function and insulin resistance. So the GPLD1 levels were negatively correlated with this HOMA1, which is the assessment. So let me see, is high good? I shouldn't go to that. I should go to Wikipedia. Oh. So glucose times insulin divided by 22.5. So this would be, the glucose is in molar units of millimoles per liter, and okay, well, if you have more glucose and more insulin, that means it's higher. So let me look at this real quick just to make sure that I have this under wraps in my head. So the more GPLD1 you had, the less insulin and less okay, well, I won't be able to parse that right now. Just know that it is negatively correlated with the insulin levels. So the less GPLD you had, the more insulin you had, and the less GPLD1 you had, the more insulin you had. Okay, so what is this telling me? This is telling me that if you exercise or if you take this chamomile flower extract, which is shown to have some beneficial effects in diabetes, that you end up having less GPLD1. So this is confusing. I thought GPLD1 was supposed to have a protective effect, but it looks like it's not. So here are the conclusions. The study suggests that endurance training and chamomile flower extract may downregulate serum GPLD1 levels in these STZ nicotinamide-induced diabetic rats, which associate with serum insulin profile. However, the results show that endurance training and CFE may not cause serum GPC4 adaptation in the diabetic rats. Okay, so what I'm getting here is that exercise is lowering GPLD1 in these diabetic rats, but in another paper, we're learning that GPLD1 is actually upregulated by exercise. So where is the disconnect here? Well, let us move to this other article, and I won't go over the entire thing. Well, actually, I shouldn't. Wait 1 second, sorry. The response of, okay. And then it was, sorry about this. So this was by these people, and then this. Okay, so this was another. This was the same group actually. These Farzad. Okay, so basically this is a fuller paper in Springer Nature that you can also read, but it basically was the same thing. We'll go to the concluding remarks. So the present study revealed that experimental diabetes increased circulating GPLD1 levels in rats. Simultaneously, we studied that the endurance. Oh yeah, so by giving rats by giving rats this toxin, it actually increases their GPLD1 in comparison to the healthy mice. So to me, what makes sense is that this GPLD1 isn't the cause of these negative effects. Rather, it is a protective mechanism that is being employed due to the fact that these rats are being damaged with this toxin. So simultaneously, we studied the endurance exercise training as an approach to investigate the alterations and regulation of circulating GPLD1. And the findings showed a difference in circulating GPLD1 levels in normal sedentary diabetic and trained diabetic rats, as well as a significant association between circulating GPLD1 levels with glycemic parameters and circulating insulin levels. Our results demonstrated for the first time that endurance exercise training restored, so this would be the better way to describe it, it's restoring the circulating GPLD1 concentrations in diabetic rats to normal levels. So we give these rats the toxin, their GPLD1 levels are going astronomically high. But if they exercise, then that GPLD1 lowers back, or I should say restores itself back to, or closer to, normal levels. So the findings also indicated that alterations in circulating GPLD1 concentrations were not accompanied by circulating GPC4 concentrations. So here they were thinking that GPLD1, if it was upregulated, then it might be releasing more GPC4, but apparently that's not the case. So further experiments are required to determine the effects of exercise training on the regulation and function of GPLD1 in diabetes. So let's step back. This is saying that the effects of GPLD1 lowering, this is explicit to their particular diabetic model that they're using. So yeah, so what I am going to do is go over, so this is the serum GPLD1 following the 14 weeks in the group. So here we have the sedentary control group. Here we have the sedentary diabetic group, and you can see it increases significantly. And here we have the trained diabetic group, where they underwent training, and then it ended up going back down to healthy levels. Or I shouldn't say healthy levels, but it was going back down. So, yeah, I mean, does this make sense to me? Who knows? What I really wish that they had was is if they had a trained model without having diabetes. So having a trained control versus a trained diabetes, and see what that correlation is like. So, yeah, if you want, you can read this paper as well. I'll leave it down in the description. But I also want to go over this paper, which is insulin resistance is associated with a compensatory, well, it's glucose and insulin regulate glycosylphosphatidyl inositol specific phospholipase D expression in the islet beta cells. So, and here I did look up, this isn't a different glycosylphosphatidyl inositol specific phospholipase D. This is specifically GPLD1. So don't get, yeah, I did look it up. And so GPLD1 doesn't seem to just be associated with the liver, it seems to also be associated with the islet cells. Now, something that I want to think about is when we take these plasma tests of these rats, we don't know where the if we're getting it from the plasma, we don't know if it's coming from the liver or these islet cells or the beta cells. We just know that it got secreted into the body. So if we are inducing, say, diabetes into a creature, then how do I phrase this? If we induce diabetes, then that is going to screw up specifically the production of GPLD1 within those islet cells explicitly, right? Or more so there than others. So could it be that the liver tries to ramp up production to compensate for the loss of activity of the islet cells? Let's read and let's learn a little bit more about this. So insulin resistance is associated with a compensatory islet hyperactivity. So if you are insulin resistant and you have way too much glucose in your blood, then the islet cells will start producing more and more insulin as a way to compensate for the fact that your other cells are resistant to sucking up the glucose. And it does that to sustain adequate insulin biosynthesis and secretion to maintain near euglycemia, or this would be normal levels of glucose in the blood. So both glucose and insulin are involved in regulating proteins required for insulin resistance, or insulin synthesis. So insulin regulates its own synthesis as well, and secretion within the islet and islet hypertrophy. So we determined that glycosylphosphatidyl inositol specific phospholipase D, GPLD here, but this is the same thing as GPLD1. So keep in mind, this is GPLD1. This is present within the secretory granules of the islet beta cells. So to determine if GPLD1 is regulated in the islet beta cells, we examined the effects of glucose and insulin on GPLD1 expression in rat islets and murine insulinoma cell lines. So this is a cancer cell, presumably, I would assume, from the beta cells of a mouse, and basically they're using it as a cell line to run experiments on. And we use cancer cells frequently for these cellular experiments because they reproduce forever, or they reproduce a lot. So we are able to we're able to have reproducibility and have a lot of different cell lines. Anyways, glucose increased the cellular GPLD1 activity and mRNA levels 2 to 7-fold in the isolated rat islets and the beta TC3 and beta TC6 F7 cells. So I assume these are the murine insulinoma cell lines. So insulin also increased the GPLD1 mRNA levels in rat islets and beta TC6 F7 cells 2 to 4-fold, commensurate with increased GPLD1 biosynthesis. So not only were the mRNA levels, or the transcription of GPLD1 was increased, but they also did see more protein. Oh no. Okay. So to determine if islet GPLD1 expression is increased in vivo under conditions of islet hyperactivity, we compared GPLD1 mRNA levels in the islets and liver from OB/OB mice and their lean littermates. So what are OB/OB mice? So these are mutant mice that eat excessively due to mutations in the genes responsible for the production of leptin. So this is a hormone that helps, this is a satiety hormone. So as you eat, you release leptin, which then convinces your body that you are satisfied. And so these mice have a mutation of that, and they need both of the mutations. This is why it's OB/OB. So yeah, it'd probably have to be in both chromosomes that are both alleles where this is taking place. And so here you can see a mouse that has these mutations, and therefore they will never, if they are given the opportunity, they will just continue to eat as much as they can, right? So you can theoretically put them on diets as well and prevent that from happening, except that's not the reason why we're doing these experiments. So in these OB/OB mice, which remember, these OB/OB mice still, they should theoretically not have the same type of screw up as the AZT, or not AZT, what would the word be? So this isn't a toxin that causes obesity. This is a gene that causes obesity. So their diabetes is going to be more natural, for lack of a better term. So this islet GPLD1 mRNA was increased fivefold, while liver mRNA and serum GPLD levels were reduced 30% in OB/OB mice compared with the lean littermate controls. So let me read this again. The islet GPLD1 mRNA, this increased a lot in the obese mice. And the serum GPLD1 levels were reduced in the obese mice compared with the lean littermate controls. Okay, well I'm going to criticize that a little bit because we do know that as you age, your GPLD1 levels are decreasing. They decrease with age, and by exercise, you can keep it up, right? So here, I don't know if this is really convincing. However, it is to me convincing enough that there is a big difference in the serum levels of GPLD1 of the obese mice. However, the mRNA in the islets for this GPLD1 is really, really, really high. So it's increased fivefold. So, but in the liver, it's very decreased. There's not nearly as much of this GPLD1 mRNA being synthesized in the liver as a result of obesity. Interesting. Now, granted, this doesn't mean that the pancreatic cells or the islet cells, it doesn't mean that these are the cells that should be doing it, right? So by producing so much protein all the time, these islet cells can actually become damaged over time. So yeah, well, I'll keep an eye on this. So these results suggest that glucose and insulin regulate GPLD1 mRNA levels in the isolated islet and beta cell lines. So these regulators may also account for the increased expression of GPLD1 mRNA in islets of the obese mice, which is a model of insulin resistance and islet hyperactivity. Interesting. So what I'm reading from this is that if you have glucose in your system and if you have insulin in your system, then those are going to greatly increase the GPLD1 expression of the pancreatic cells, but it's going to reduce the expression of GPLD1 in the liver cells. So maybe this is what is, how do I phrase this? Maybe this is what is causing some damage, right? So maybe we need to focus our attention into expressing GPLD1 in the liver, find ways to cause the liver to produce it, which, you know, your liver, much larger, much more capabilities at producing these, a lot of other proteins, right? A lot more reparative. The liver is really excellent at repairing itself, right? So being able, I mean, you can even have a chunk of it removed and you can still survive pretty well, and I think it regenerates a little bit too. So if the attention is focused on to the liver and protecting the pancreas, that might be beneficial. I wouldn't, yeah, so how do I phrase that? So going back to the mouse models where they ended up destroying a lot of the pancreatic cells with the toxin, the GPLD1 levels were high. So if the GPLD1 levels are high, where is that coming from? It could be coming from the liver, or as a compensatory mechanism. You know, this is one of the things that I dislike about biology, is that there are so many cycles and there's so many self-regulatory mechanisms that it's really hard to make any definitive statement, because anything you say about a protein in the body, there's usually a negating protein somewhere else in there that could cause the opposite effect. So, but I'm trying to create a model which would make sense. And to me, what makes sense is GPLD1 is a protective enzyme. And but what I mean by that is that given certain damaging things happening to the body might produce more of it to help protect the cell. But granted, sometimes high expression of something in low expression of something can confer protective effects, but sometimes when it's very high expression, that ends up making damaging effects, and that is true. Okay, so this is what's happening in my head right now, is sometimes, let's say you have a defense protein that is being synthesized in result to damage of the system. So if you have a little bit of those defense proteins, and I wouldn't be able to specify which proteins I'm thinking of right now, but if you have low levels of them, that convinces the cell, oh, I need to repair myself, I need to fix this damage. However, if you have so much damage to those cells, you end up producing a lot of those same proteins, a lot of those protective proteins. But if you produce too much, then that actually convinces the cell that there is too much damage and therefore I should kill myself. So maybe that is the strategy that is being employed here, is that when you are, I guess when you are inducing, let me see, this was, so the liver mRNA and the serum GPLD levels were reduced in these obese mice. So it wasn't just the liver, it was also the serum. But regardless, it could be a protective mechanism, and the pancreatic cells might be expressing so much of this GPLD1 that it's no longer a protective molecule. It might actually end up becoming an apoptotic molecule. So I will, something that would make me think that way is if this GPLD1 plays a role in apoptosis. So let me bear with me for a hot second. Let's look up Scholar, and let us look up GPLD1 and apoptosis. So GPLD1 apoptosis. Oh, so here we go. 10-phenanthroline, which is a GPLD1 inhibitor. This has protective effects against inflammatory and/or stress-induced apoptosis. Okay, so this is telling me that if you have a drug that suppresses GPLD1, it can prevent apoptosis. So remember, apoptosis is programmed cell death, right? And apparently there is a molecule, a drug, that can inhibit GPLD1 and as a result prevent apoptosis from happening. So keep in mind, apoptosis is extremely important. You need to have the ability to kill cells so that they do not become cancerous, or that they do not continue to multiply after they've received significant damage. So very, very, very frequently, it is important for a cell to understand when it has received too much damage or it does not belong, and then it undergoes programmed cellular death. And here, it seems that a GPLD1 inhibitor is able to have a protective effect against apoptosis. And this has been associated. Let's see. So an apoptosis-related gene signature for the prognosis of hepatocellular. No, I don't. So remember, frequently we want to prevent apoptosis in nerve cells because they're not very good at duplicating. You can create new axons pretty readily, but the duplication of neurons tends to be pretty slow. So actually, I don't think the neurons themselves can multiply. I think it has to be some form of glial cell or stem cell or some sort of multiponent cell within the brain or within the nervous system that has to actually go in and recreate a nerve. So let me look at GPLD1. So these are enzymes and anabolic and catabolic chemical reactions. And so some studies have focused on the inactivation of these enzymes in a range of malignancies. Drugs like pegylated arginine deaminase. Here we have 1,10-phenanthroline, which is a GPLD1 inhibitor. These have already been reported to act as sensitizing anti-cancer agents. Okay, so maybe that was not correct. Protective. Okay, so I take back what I said earlier. Here it looks like the protective effects against anti-inflammatory and stress-induced apoptosis that was not associated with the GPLD1 inhibitor. So the GPLD1 inhibitor that was a sensitizing agent. With the exception of they are already known targets for neuroprotective drugs like temporal or iron chelators. The functional effects of pharmacological modulation of other enzymes remains to be clarified, so some studies have focused on the inactivation of these enzymes in a range of malignancies. So these drugs have already been reported to act as sensitizing anti-cancer agents. So inhibiting GPLD1, well, to me this sounds like GPLD1 is inhibiting apoptosis, and so by inhibiting GPLD1, you end up increasing the likelihood for apoptosis. So I don't know. I don't know what's going on there. Let's see. So small molecule alteration of genes. You know what? I don't think I'll be able to find anything here. So what I'm going to do is leave at that, probably. So what do I want to say about all of this is it appears as though it is extremely, extremely difficult to figure out how this protein needs to be differentially regulated in order to work properly or have the benefits that we want to have. But what does appear to make sense is if GPLD1 is expressed in the liver, And maybe as long as it's not being expressed highly in the beta cells of diabetics, then it ends up being beneficial for the body as a whole. So if it's produced in the liver, that seems to have some benefit. And so in this, for diabetes, it appears as though the expression in the serum and the liver seems to be lower if they have diabetes induced from eating too much. So that eating seems to cause, so the high levels of glucose and the high levels of insulin seem to increase the GPLD1 expression of the pancreatic islet cells. Okay, but that does not seem to be causing, or I should say, that is not causing the upregulation in the liver cells. So perhaps there is a net negative by having diabetes in the production of GPLD1. This is explicitly overeating diabetes, so this would be type 2 diabetes. So going back to these papers where they actually kill the islet cells. Okay, so bear with me here. So in this situation where they are actually killing the islet cells, I would presume that would be lowering insulin. So let me go back to this. So here we're saying that the GPLD1 in these so here are the results. So GPLD1 levels were negatively correlated with insulin levels. Okay, so maybe this is it. Maybe the serum GPLD1 levels, the GPLD that comes from the liver, or maybe some other organism or some other tissues, but predominantly comes from the liver, this gets, I should say, insulin prevents it from releasing. Yes, okay, so having so if you have very low insulin levels, you end up releasing more GPLD1 from the liver into the body. If you kill the islet cells and you lower insulin in the body, which is not necessarily a good thing. Insulin is important in the body, right? So you end up, the liver gets the signal somehow that I need to produce more insulin. Or sorry, I need to produce more of this GPLD1. So instead, okay, but the pancreatic cells, if there is low insulin, they produce less GPLD1, right? So even by killing, so remember, if you have these insulin cells in the pancreas and you kill a bunch of them, that insulin, they'll produce, or they won't have the ability to produce as much insulin, and so that actually ends up screwing up the signaling even between these cells, and that could lower the GPLD1 and insulin expression of them. So, and then we have insufficient insulin responses as a result, but that would also mean an insufficient GPLD1 levels within the pancreatic cells, but the liver would end up producing a lot more GPLD1 since there is no insulin. Well, that's fascinating. Now, thinking about this, does this mean that GPLD1 is this a protective effect that was evolutionarily conserved? So if you can think about this with me. So perhaps throughout history, it would happen that some people would become insulin resistant, but they would have type 1 diabetes as a result of their beta cells being destroyed. And then they wouldn't produce sufficient GPLD1. But the liver would come in and realize that there is not enough insulin being produced. So the insulin can sense the fact that there's not enough insulin going around, and as a result, and this could probably be to low levels of glucose in the hepatocytes. The hepatocytes are the main cells in the liver. So if they aren't getting enough glucose, or if they're not getting enough insulin, one or the other, they get into this mode of producing more GPLD1. That's fascinating, which would also indicate, bear with me, people who fast and people who do not eat a lot, or they will go through periods of not eating. They will have periods of lower I shouldn't say lower blood sugar, because your body does undergo some form of homeostasis, but this would convince the liver there's low insulin and there's low glucose, or lower than normal, and therefore I need to produce. Now, well, in the type 1 though, the glucose levels can get very high, right? So maybe it's not glucose, maybe it is explicitly insulin that is convincing the hepatocytes whether or not they synthesize GPLD1. I'll have to look at a map of these biochemical or signaling pathways to make sense of it more. So this would lead some credence to the belief of people who fast, people who do not eat for 18 hours a day, but then will eat later in that day. This would lead some credence to believe that people who do undergo fasting will end up having a higher expression of GPLD1, and this higher expression of GPLD1 ends up benefiting or providing some protective effects to the rest of the body. Now, if someone has their beta cells damaged, that's not good, right? So they're probably not going to, someone with damaged beta cells, they're probably going to die quicker, right? So it's probably a larger effect than what the benefit of the GPLD1, right? So the effects of having high blood sugar all the time, all that damage, is probably going to be greater than the protective effects of the high GPLD1, right? So, but the high GPLD1 without diabetes will probably be very beneficial. So, yeah, I think that's the most appropriate model for all of this. And so, with that, I think trying to make sense of, or at least model this particular protein that way, might make more sense or I should say, it might help with the understanding of the galectin-3 and the galectin-3 binding protein, how they are expressed. Yes, I think so. So what I'm going to do is I'm going to quit here, and then I'm going to have another video tomorrow about the galectin-3 and the galectin-3 binding protein, which with yesterday's video, I went over those proteins and said that they might have a protective effect, but when I did more reading, it appears as though those are actually pro-inflammatory, which, remember, inflammation is there to protect you as well. But it seems that galectin-3 and galectin-3 binding protein have this pro-inflammatory effect, right? So they don't seem to be good at repair, they seem to be good at inflaming. But what I'm going to do, or what I'm going to suggest, is go over how GPLD1 and how the galectin binding protein, what is that link? Because if that link is heterogeneous throughout the body, and dependent on whether it's in the tissues or in the serum, that might help us understand a little bit better what the hell is going on and how GPLD1 exerts its neuroprotective effects. So with that, I think I'm going to head on out. But thanks for listening to my meanderings, and hopefully I will see you tomorrow.